So now we're finally moving into another system of the body. When we look at this system, we're going to see that a couple of chapters are going to be concerning our circulatory system, our heart, our blood vessels. So one of the things that we're going to start out with for Chapter 18, in this circulatory system, we're going to start out with the blood. This is, I think, the actual picture from your textbook. I kind of liked it. And it's showing a red blood cell. And then, even though it says the white blood cells in blue, it's not kind of showing up because of this LED being so bright. But this is going to be the white blood cells. And then we're looking at some platelets, which are kind of interesting. And then we've got some filaments that are extending. Believe it or not, that's a clotting protein that travels around in the blood, and it's very important in that clotting process, meaning something as simple as, say, a paper cut, the body's got to make sure it closes that area to the outside environment. So in looking at blood, part of the circulatory system as a whole, there's going to be a lot of stuff in blood. Has anybody looked at the chapter yet? Okay, so look at it, all right, because it's pretty amazing, all of the stuff that's in it. When we say the circulatory system, like I said, we're going to break it into heart, we're going to break it into blood, we're going to break it into vessels. So, some people refer to it as the cardiovascular system, okay? But the part that we're going to look at, it's going to have that word heme associated with it. So when you hear heme, let that make you think about blood. So the study of blood is hematology. Interesting topic. I would not personally want to sit around all day and look at slides of blood, but it's important. I mean, like if I'm having a biopsy or something done, or they've got to check my blood for something, definitely want the person behind that microscope knowing what they're doing. For blood, okay? Well, the purpose, the functions, what it's going to do. We need it for transport, we need it for protection, and we need it for regulation. Blood. Now let's think about this for a second. When we think about the blood in the body, leaving the heart by way of the aorta, going all the way around the body, coming back to the heart, you're probably going to think about this vascular system, and you're probably going to think about it in reference to it looking something like this, right? Where you're thinking of arteries in red, veins in blue, and then big ones. Is that correct? yes and no. These are definitely these larger vessels that are getting represented here. These are definitely the transport vessels. All right? It's where we're carrying everything that the blood is going to carry. When I say everything, trust me, it's a lot, okay? Because I'm going to carry nutrients. I'm going to carry waste. I'm going to have red blood cells. I'm going to have white blood cells, the ones that are for protection. I'm going to have antibodies, which are for our immune system. Okay? We're going to see the platelets. I'm going to need materials to regulate, especially buffers. Can you close the door for me? Thank you. What is a buffer? Marlisha? Now, keeping it in balance based on what type of scale do you?
The pH scale?
The pH scale. pH tells me the numbers for what in a solution?
Acidity.
The acidity, the acid, or the base. All right? Do you remember what number neutral was?
7.
7. Okay? Anything below that, acidic. Anything above it, basic. Right? Now, do you think the blood should be more acidic, neutral, or basic? It definitely needs to be close to neutral, with it being
More basic.
just a smidge over 7. But then other areas of the body will actually be acidic. Others can be basic. The blood as a whole needs to stay kind of close to the midline. But wait a minute. If one of the jobs is doing the carrying of nutrients, picking up of waste, somewhere there's going to have to be an exchange of these materials. The exchange of these materials does not happen at this large level that veins and arteries are named for. It happens at the level of a capillary. The capillary beds. Nobody really knows how many we have in the body, mainly because they're microscopic. It's believed that we have millions of them. So I have to make sure that materials that are being carried in the blood, nutrients, for example, that's going to be materials, for example, like my electrolytes. And I need to get those electrolytes to the cells that need them. For example, muscle and nerve. So the exchange is going to occur, but at the same time, I'm going to pick up waste. The waste, of course, big part of it, is that byproduct of cellular respiration, which is what gas? Carbon dioxide, CO2. Now, CO2 into a fluid environment, i.e. the blood, that contains a whole bunch of water, well, Any idea what gets produced when CO2 gas is in a water environment? How many of you drink sodas? Every time you drink a soda, you're drinking something called carbonic acid. It's a weak acid. However, it's an acid from CO2 that in our bodies, when it gets into the bloodstream, needs to be buffered. Because it's acid gets into the blood, does that not make the blood more acidic? I got to buffer that. That's just one of the very many things that the blood is going to have to do. So, important. Transport, protection, regulation. In part 1, when you were discussing the types of tissues in the body, do you remember that blood is a liquid connective tissue? All right? So blood is actually connective tissue. It's just in the liquid form. It is going to contain cells that keep their own structure. It's going to have an extracellular matrix. If you remember about connective tissue has a matrix. In this case, the matrix is plasma. At any given time, we can have roughly 4 to 6 liters of blood in our body. Plasma, when you separate blood out, has a clear light yellow color to it. And then our formed elements. We're going to find out what that is. Because they have kept their structure while existing in the blood, they separate out. This would be cells, for example, red blood cells, white blood cells, and then a cell fragment, which are our platelets. Platelets are not a cell. They are a fragment of a cell.
What are they missing?
They actually are just broken off pieces. They don't have like all the organelles, like the nucleus, the endoplasmic reticulum, that sort of thing. They are simply pieces that have broken off from what we call the pluripotent stem cell. And it's basically just the plasma membrane with materials in it. In our blood, those formed elements, a cell, the cell fragments. In a blood smear, like if somebody were to take a drop of blood, stain it, because they do, that's how they get to see different cells. In a fresh drop of blood on a slide, it's really cool because you see the red blood cells very well. But it's white blood cells that you're not going to see very well. So they use different staining techniques to be able to see the white blood cells. When you look at it under a scope, that becomes important because in the normal situation,